Good day, friends, and you're welcome back to the channel. Today is part 2 of our series on the cultivation of viruses. Now, we have asked in an earlier video what are the methods that can be used to cultivate viruses. We said animal inoculation, we said tissue culture, and some other methods. Now, for this video, we're going to be focusing on the third method, or on the second method, sorry, which is the use of an embryonated egg. The use of an embryonated egg to carry out cultivation of viruses. This is important to note. Now, this practice was started as far back as in 1931 by a scientist which was called Good Pasture. Good Pasture was the name of the scientist. He's the one who designed this method of cultivation of viruses that uses the embryonated chick egg to culture or cultivate viruses. Now, this method depends on the kind of virus which is to be cultivated will determine the way this method will go. Now, most often used is the chick embryo, and it's usually a chick embryo that is within the ages of 7 to 12 days. Now, we should remember that every time when we want to cultivate viruses are usually cultivated in a medium that has rapidly growing and rapidly dividing cells. That is why the chick embryo or the embryonated chick egg can be used. So this is important to note. Now, the process involves, first of all, you sterilize the shell. The shell of the chick egg is sterilized. Then after it's sterilized, a sterile bore is used to create a hole, bore a hole into the chick egg through the shell. Now, when this hole has been made, the viruses are inoculated into the chick egg, and when they're inoculated, then the chick egg is incubated at 36 degrees Celsius and is incubated for 2 to 3 days. After 2 to 3 days, the egg is brought out and the viruses must have reproduced and replicated within the chick embryo. So this is the process. And after this process has been done, the chick egg is broken and then purification is done to separate the cells of the egg from the viruses which have replicated within this cell. And this is quite important to know. Now, it's also important to know, like we know, that the chick egg has very different parts, many different parts. And it's important to note that viruses can be grown inside all of these different parts. That the parts of the egg in which viruses can be grown include the allantoic cavity, the yolk sac, amniotic sac, and chorioallantoic membrane. All these are parts within the chick egg, and in all these different parts, the viruses can be cultivated, viruses can be reproduced and replicated in all these parts of the egg. Now, what are some of the advantages of using an embryonated chick egg to cultivate viruses? Some of the examples include that it's a widely used method, is less laborious, is easy to do, is cheap and effective, and then eggs are readily available. So the process can be run quite easily. Maintenance is easy, and the process involves a sterile process. Now, this sterile process is helpful because it reduces the contaminants of bacteria in the whole of the process. So these are the advantages of using the embryonated egg. Now, the disadvantage of using the embryonated egg to cultivate viruses is that many a times different viruses replicate or reproduce in different parts of the chick egg. So sometimes it's difficult to know into what part of the chick egg should a particular virus be inoculated for such a virus to replicate. So this is important to note. So this is the discussion on the cultivation of viruses using embryonated eggs. This is important to know, and this is a method of cultivating viruses. Thank you for listening to this video. Please like our content, share our content, and do have a wonderful day.